Thank you very much, LabRoots, for giving me the opportunity to present today about the landscape of pharmacogenetic resources. I will focus on the Pharmacogene Variation Consortium, what we offer to the pharmacogenomic community and how we fit into other resources and what we provide to the community research and translational wise to further research and implementation. So before I really get to FarmVar, I want to take you back about 20 years to the year 1996. This is where cytogram P450 nomenclature was first published. And back then, a group of experts got together as they realized that variation in pharmacogenes is really a challenge as many methods experienced a revolution, and resequencing and gene characterization became really what labs were doing much more easily and fast. More and more variants were discovered, and so the need to really find a common language to catalog these variants in a way we could all understand was really central. And so they encouraged scientists to speak the same language worldwide and came up with a nomenclature that became known as the STAR nomenclature. And so because these variants were becoming more and more, and in 1996, the first paper was published on cytogram P450-2D6. And then 5 years later, around 2000 or 2001, this nomenclature was expanded to other cytogram P450s. And it was shown in the last paper that kept track of these really in paper form. They had a list of over 25 pages or so of variants that was not feasible and sustainable over time. But then there was this internet, which of course provided the perfect platform to do so. And this is when the cytogram P450 nomenclature page made its debut on the internet, and that was back in 2002. And that was under the leadership of Magnus Engelmann Sundberg at the Karolinska Institute. And so as you can see here to the left, this is the catalog of cytogram P450 genes for which this website kept track of and built this nomenclature over the next 15 years. And the pharmacogenetics and genomics community has become reliant on this centrally run database. People submitted their novel variants, and it was really the go-to page to inform yourself and pull the resources of pharmacogenetic variation. But then in 2017 is when Magnus Engelmann Sundberg decided to move on to other research endeavors. He was looking for a new generation of experts to take this over and actually really lift this nomenclature page up into a new century and into a new database. And this is where myself and others, other experts from the PharmGKB and other experts in the field, again, came together and stuck our heads together to see how can we take that and build a better database to serve future generations in pharmacogenetics with what Magnus has built before. And this is where the human CYP allele nomenclature database was transitioned into the FarmVar database, or to the Pharmacogene Variation Consortium. And as you can see here on this slide, we work closely together with the PharmGKB, another well-known database for pharmacogenetic variation. Before I go further ahead, I also really want to give you a quick run through of what the pharmacogene STAR nomenclature really is. So what you see here on the left is how it's written. So you first have the species. So this is for humans. You have the superfamily gene name, which is CYP or CYP for short. You have the family, which in this example is 2, the subfamily, which is D. Then you have the isoform, which is 6. And then the allele, the different variants, is what is that star number. So you can see here, in this example, it's the star 5. So there are at least 5 different variants, and these are all different haplotypes for this gene. So for 2D6, we are now up to 141. So this is an example of a highly variant and highly polymorphic pharmacogene. And so PharmVar holds mostly cytochrome P450, so this is what we have inherited from the original nomenclature site, but we are also expanding to other clinically actionable pharmacogenes. And so far, we have introduced a nomenclature for NUT15 and more recently on DPYD. And as I will show you, we are also starting to work on our first transporter, SLCO1B1, later this fall. Now, there are also other web-based sites for other genes, namely TPNT, UGTs, NATs, and ALDH nomenclature. But yet for others, there are only recommendations. There is no standard nomenclature, no centralized site that keeps track of everything. And so the star nomenclature for these might be very loosely applied and used in the literature, which can be really difficult to follow up and keep track of. So why are haplotypes important in the first place? So this is what I want to show and demonstrate here on 2 examples for cytochrome P450 2C19. So in the top half of this slide, you see an example of a diplotype we call star 1 star 2 and star 1 star 35. And so you see 2 SNPs here in red. These are both SNPs that confer alternative splicing. And so on the allele we call star 2, both SNPs are present. In the star 35, however, only one of these is present. Now the SNP that is typically tested for is C681. Now you can easily see, should this SNP not be tested, well then you are not identifying the star 35. And what that means is that this allele would default to a star 1, and then your genotype would not be accurate, and the phenotype you predict from this subject would not be correct either. So knowing which SNPs are together and can occur on the same haplotype is rather important, because that might direct you to what is important for testing. And it also allows you to gain more understanding of these SNPs that are tested in an assay or by a company, to see what this test menu might miss and which alleles you might not see and which might default to a star 1. Now in the bottom example, we have an allele that is called a star 4. And here there is an A to a G SNP, and that affects the translation start codon. Now this SNP is often accompanied by the SNP shown in blue, which is in the upstream region. And if that SNP occurs on its own, it actually increases expression levels. Now these 2 SNPs can occur in cis, then it makes a star 4 of 001 subtype, or they can occur in trans. And while either diplotype is translated into the same phenotype in this example, you could easily imagine that if that more rare SNP in red that defines the star 4 allele is not tested, you would really end up with the wrong genotype, because that star 4 is a no-function allele, while the SNP shown in blue leads to increased expression and is therefore signifying an increased activity allele. So I hope these two examples really exemplified how important it is to know which SNPs occur together and what the functional consequence is. So this here is the Pharmacogene Variation Consortium landing site. And if you go to the tab Genes, you will get to a gene list from which you can pick the gene of interest. And on the next slide, I'm showing the gene page for cytogram P450 2D6. And on this site, or each gene page, you find really a series of important information and features, some of which I have highlighted here with the yellow arrows. So one I really want to emphasize and point out is the one underneath the top gray bar where it says important gene documents. So here is a README, a changelog, and structural information files. I encourage everybody to read because these files contain very important information for the gene, for the website, how we list things, what standards we use, and also give you examples of the features of this gene page. So in fact, a lot of questions I receive, I can answer by, oh, this is all detailed and explained in that README file. So I really encourage everybody to check out these files. So especially for cytogram P450 2D6, which has a high level of structural variation, we have summarized a lot of that information in the structural variation document, which might be very helpful for your work. Then we have a number of features. So you can choose different reference sequences to have the variants are mapped to genomic transcript reference sequences, but also both genome builds. And in the case for CYP2D6, also a legacy sequence. You can also choose different count modes, have them mapped to the ATG being +1, which is very helpful just to toggle between these count modes. You can also create custom views using filter options. And what we also offer are the CPIC clinical allele function assignments as issued by this group. And we have introduced core allele definitions, which are essentially is a rule-based system to collapse all sub-alleles we are cataloging into a major allele definition, if you will. And finally, we also assign haplotype evidence levels. So this really gives you a feel for how much evidence there is to support a given haplotype. And this is not to be confused with evidence for function, but really a quality information to tell you whether a haplotype indeed exists or may not exist in the form. We are posting it here. I want to quickly give you an overview of our comparative allele viewer or CAVE tool. So this is a pretty nifty tool where you can choose the alleles you would like to compare on a selection pad, which is shown on the left. And then on the right, it shows you a graphical comparison of these different allelic variants. And so you can see all these blue squares, the little pill sign, the PharmVar logo. These indicate SNPs that are unique to an allele. So in this example, there are two SNPs highlighted by this logo, a star 4 SNP and a SNP you can find in the star 100. And so if you find these SNPs, you have one or the other allele defined. But you can also see in the top row for the SNP that says 100 CT that this particular SNP can occur on numerous different variants. And so if you see this particular SNP, you cannot really tell whether you have a star 10, 36, 49, and so on. And so you need to do additional testing in order to really drill it down to which allelic variant you have. So seeing which SNPs occur on which allele in a graphical form is very helpful, in addition to have everything just tabulated. So as I already mentioned before, we have now also genes that are not cytochrome P450 genes. So you see here, not 15, which we have launched a little bit more or almost 2 years ago now. DPYD we have launched earlier this year, and SOC01B1 will start work in this year. Describe what we do, how we do it, what kind of features we have built, and how we interact with other landscapes. But we also have had so far 2 papers, what we call gene-focused papers, in a series we are building. There was the first one was on cytochrome P426. The more recent one was on 2C19. And we are working on one for 2B6 right now. And these gene-focused papers give you really a comprehensive overview of the variation of these genes, what PharmVar is doing, and everything you need to know about these genes. I also want to highlight that we are offering templates for authors to use to describe genotyping methods, report alleles and diplotype frequencies. And we have devised these tables because it is sometimes really, really difficult, for example, CPIC authors or PharmGKB curators to tease out that information from publications. And so we encourage everybody to use these templates to be really clear cut on what was tested, how was a genotype translated, and so on. So that would be really helpful. So how do we fit into with other resources, and who are we collaborating with? So first of all, clinical pharmacogenetic test reports often rely on the star allele nomenclature. So if a test report uses that nomenclature, of course, of them, allele definitions. So helping and working with the AMP, which is devising recommendations for clinical testing. We're also working with a group that is called the GETRM, or the Genetic Testing Reference Materials Group, because it is, of course, very helpful to have reference samples for your testing in the clinical arena, but of course also in research. Then we are working very closely with the PharmGKB, because it is absolutely important that we use these allele definitions in a standardized way across all these different resources. So there is absolutely no questions about what does this allele mean or that allele mean. So standardized usage of these terms and allele definitions is key. And then, of course, the nomenclature is also used by bioinformatic tools to develop algorithms that can call these star alleles from testing materials. And all this together is, of course, instrumental to eventually inform CPIC guidelines. And as you will see, they are using PharmVar allele definitions, if existing, to inform the guidelines and the translation into phenotype. So for the PharmGKB, this is probably the best known database for pharmacogenetics knowledge. And so you find information for annotated drugs, curated pathways, clinical guideline annotations, drug label annotations. But I really want to highlight at the bottom of the slide, in the middle, you see gene-specific information tables. This is a plethora of information, how to translate, for example, a diplotype into a phenotype. So if you're not familiar with these reference tables, please go and check them out. And then, of course, the PharmGKB is also known for their wonderful pathways they are putting together. The Clinical Pharmacogenetics Implementation Consortium is now 11 years old. They have identified that it is really a barrier to the implementation of pharmacogenetics, of how to interpret what is a physician to do with a genotype when it is available. And to really address this barrier, the CPIC was formed to provide guidelines that enable the translation of genetic laboratory tests into actionable prescribing decisions. And so it is important to understand that the CPIC does not recommend whether testing should be done, but it is designed to help clinicians understand how an available genetic test result can be used to optimize a drug therapy. So what you see here is table 1 from a CPIC guideline. And all these tables 1 are very similar in all these guidelines. And so you see the phenotype definitions like ultra-rapid, normal, or intermediate metabolizer. For cytogram P450 2D6, that is the activity score system that is used to facilitate the translation of genotype into a phenotype. There is a brief description of the genotype. And then to your right, you see examples of CYP2D6 diplotypes. And again, you see here the star nomenclature. And these go back to these definitions that are listed in FarmVar. So we are working very closely with the CPIC, again, to standardize the usage of these star alleles very consistently among the CPIC, PharmGKB, and FarmVar. And this is also a reason why we have fairly high standards before we define an allelic variant, to make sure that it is the best possible definition, because we know that CPIC might eventually pick up an allelic variant and is basing clinical recommendations on an allele. Then clinical test reports use star nomenclature. So these two examples are very generic examples of reports you might see, and tests might report star alleles in a table form, or as you can see here on the right side, also list the genotype and then give you a little range of where the phenotype might be. And so again, it is very important that unified nomenclature is used for these test reports, because otherwise it is really impossible to interpret these, especially across providers, as patients might leave one institution or switch physicians. So there is definitely still a lot of work to do to make that more unified and standardized. Then, of course, there are so many different clinical labs out there, and everybody is doing testing slightly different by method or what they test for. And so the Association of Molecular Pathology has started to come up with clinical genotype allele selection recommendations. They are using a two-tiered system. So tier 1 is a minimum panel of alleles that should be tested. Tier 2 is an extended panel of variants. And their selections for these tiers is really based on how frequent these are, whether an allele occurs in just one or more populations, whether there are testing materials available, and so on and so forth. And they also use, of course, FarmVar allele definitions if they are available. And so for cytogram P450 2C19, they have already published recommendations, and they also have published recommendations for 2C9, and recommendations are underway for cytogram P450 2D6. Moving on to the GETRM project. So this is housed at the CDC and is tasked to develop reference materials for PGX testing. This is, needless to say, very important for clinical laboratories, but of course, having good reference materials is also important for anything going on in research. So they collaborate with the Corel repositories because these DNAs are widely available, and these groups enlist commercial and academic laboratories to perform PGX testing. Each sample is tested by multiple platforms by different groups to come then up with a consensus genotype. And they had two landmark papers before, and then a third paper was published last fall on CYP2D6, where 42 new samples have been added to the list, many of those containing rare alleles and unusual genotypes. And this is what you can see here in table 4. And the highlight also is that these consensus genotypes are reported out as a star nomenclature. So again, emphasizing that you do need a resource that keeps track of all these star alleles and suballeles and so on and so forth, because all these different projects and groups rely on it. Then there's also tool and algorithm developments to interpret genotype data. So there are a number of commercial platforms which also provide calling tools for these star alleles. And then there are also independent academically developed tools to interpret, for example, next generation sequencing data like Astrolabe, Stargazer, Allele, to name just these 3. And to your left, you see roughly how that works. So in that red box is the allele definition. So these are tables that go into these algorithms. And then you have your genotype data in different formats. And then the algorithm is doing its thing and is reporting out these diplotypes based on the allele definitions that it received as an input. And so you don't need to be a bioinformatician to understand that the better and more high quality data the allele definitions are, the better are these interpreted diplotypes and algorithm spits out. So again, the more allele variants we are able to define and the more granularly we can define them, the better will these tools work to call these data and provides reliable diplotype calls. And then here on the last slide, I want to briefly introduce the PharmCAT Initiative. So the PharmGKB, together with CPIC, realized that it would really be terrific to have one pipeline that uses genotype data, which is fed into the algorithm, then use the FarmVar allele definitions, CPIC clinical recommendations, so that a final report takes everything together so that you have one pathway and one pipeline from the genotype or sequence data to a final report for a patient. And so I hope I gave you some insights of how complex it is to really make precision medicine work. So we are all really in this together. And although I put PharmVar here in the middle piece, there are so many puzzle pieces that have to come together, whether it's basic research, medical research, education, the PharmGKB, PharmCAT, PGX testing, physicians, to make this happen for the individualized, for the patient to receive an individualized drug prescribing and move this precision medicine forward. And with this, I would like to thank you for your attention, and I'm happy to take any questions.